Start my presentation by looking at a quick backdrop of the genetic bioweapons industry. And I think the world first became aware of the fact that there was such a bioweapons industry when a gentleman by the name of Dr. Leonard Horowitz published a book called Emerging Viruses in 1996. Emerging Viruses, AIDS and Ebola, Natural or Intentional. That was the title of the book. And in this academic treatise, and he's a virologist and a vaccinologist, he proved that AIDS and Ebola are manufactured viruses engineered in a laboratory. His academic treatise has stood the test of time. It has never been refuted. And we know that the way academics stand and fall is on being presented and refuted. This has never been refuted. And the genetic bioweapons industry is the most covert industry on planet Earth, but it exists. I would like to go further by saying that William Cohen, the former US Secretary of State in 1997, Cohen referred to the concept of an ethnic bioweapon as a possible risk. In 1998, some biological weapon experts considered such a genetic weapon plausible. And you know when they say that, it means they've already produced it and they're stockpiling it and waiting to use it. And that research was being done on how certain substances can influence human genes. That was 1998. 10 years later, in 2008, the US government Congressional Committee sat under the title, under the label, Genetics and Other Human Modification Technologies. A new kind of arms race, people, a new kind of arms race is happening. And somebody, and in that committee said, there are attempts to manipulate human genetics in ways that would horrify us. And so that was 2008. Let me go a little further and go beyond William Cohen. And we want to introduce you to somebody called Dr. Francis Boyle. Very, very significant gentleman. I think we have a photo of Dr. Boyle that we could put up. Dr. Francis Boyle is an international law expert. He's also an expert on genetic bioweapons. And he was a gentleman who framed the Anti-Terrorism Act. It's called the Bioweapons Anti-Terrorism Act, or BWATA. It was framed in 1989, signed into law in 1990 by President George W. Bush with unanimous approval from the US Senate and the US Congress. And so if there is a Genetic Bioweapons Anti-Terrorism Act, it means obviously there must be a genetic bioweapons industry. And I want you to know, people, that on this earth, 22 nations on planet Earth have what we call BSL-4 labs. A BSL-4 lab means biosafety level 4. Biosafety level 4 is the highest level of biosafety. And in these labs, they make pathogens, deadly pathogens. I also want you to know that 16 nations on planet Earth stockpile genetic bioweapons. 16 nations stockpile. They have them in their armory. There's a new arms race going on. And I want to give you a quote from Dr. Francis Boyle. Dr. Francis Boyle, the framer of the Anti-Terrorism Act, says this, international law expert, bioweapons expert, SARS-CoV-2 is an offensive biological warfare agent made in a lab and engineered with gain-of-function properties. He said that in February 2020. I have quoted this in a documentary I did. It's on YouTube called Watchman What of the Night Part 1, where I spell this out very clearly. Wow. And he has come to the conclusion, and this is going to shock you, amen, put on your seatbelts, this is going to shock you. He has come to the conclusion that SARS-CoV-2 is a tripartite chimera, and it is composed of one, SARS, the original SARS, and he says SARS is already weaponized, and then that SARS virus was enhanced by gain-of-function properties, and step 3, they were all then genetically recombined with an HIV virus. That's the virus that causes AIDS. So a tripartite chimera. If you think I am kidding, I have some notes here that I can refer to. And I call them smoking gun 1, 2, and 3. Francis Boyle, quoting from an article called Antiviral Research, February 10, 2020, written by 3 French scientists and 1 Canadian scientist from Montreal, said the Wuhan coronavirus genetic analysis may provide a gain of function to 2019 novel coronavirus. The Wuhan coronavirus genetic analysis may provide a gain of function to 2019 novel coronavirus for efficient spreading in human population compared to other beta coronaviruses. He says that is a smoking gun. It was genetically engineered for efficient spreading in human population. Wow. That's recorded. Gain of function, if you don't know what gain of function means, gain of function means genetically engineered to be more lethal, more virulent, more infectious, and to mutate or adapt more readily with slight increases in immune selection pressure from the population. It's also called accelerated viral evolution. And I saw Dr. Gill refer to that in his campaign about, and he used the colloquial parlance, just so. Viruses don't just so jump from animal to human. Smoking gun number 2, UNC, and that's the University of North Carolina, don't be surprised. The University of North Carolina labs at Chapel Hill brought a team together to do an experiment. And part of the North Carolina University team was, they were joined by a gentleman from China called Dr. Zheng Li Shi. Dr. Zheng Li Shi. He was from the Wuhan Institute of Virology and the Chinese Academy of Sciences. And in fact, Wuhan gave a grant to the University of North Carolina so that scientists could be involved in this experiment. They imported SARS cells, SARS viruses from the Fort Detrick labs. And for those of you who know geopolitics and covert geopolitics, very necessary for any Bible prophecy teacher or any pastor in this day to know, amen, the Fort Detrick labs are the US's main BSL-4 labs where they research, stockpile, and test genetic bioweapons. And so this team, given a grant by China, were working on increasing the pathogenicity of the SARS samples that they got from Fort Detrick. Smoking gun number 3, research at the Australian Animal Health Lab was funded by the Chinese Ministry of Science and Technology, along with the other two I mentioned, the Wuhan Institute and the Academy of Sciences from China. And the experimental goal was to coalesce the initial weaponized SARS virus, enhance it with gain of function properties, and then coalesce it genetically to an HIV virus. Amen. Therefore forming the tripartite chimera. And so I'm listening to you where these experiments took place. Let me go a little further. Smoking gun number 4. Indian scientists did an analysis on the SARS-CoV-2 virus, and they said they definitely found elements of the HIV virus in the genomic sequencing. So this was confirmed by a team from India. And let me mention to you that HIV is a retrovirus. Retroviruses use mRNA to inject their stuff into the cell. And when the mRNA is injected into the cell, there is something called retrointegration that takes place. And that means that the mRNA is converted back into DNA, and then it coalesces with the chromosomal DNA of the host. So retroviruses like HIV carry an enzyme called reverse transcriptase, which changes mRNA back into DNA so it can coalesce with the genes in your nucleus. This means that when they said it's an mRNA vaccine, and they said it will only go to the ribosomes and form proteins, that's not true. Even it will be reconverted to DNA and affect your human genome. Wow. So Francis Boyle says this: the most dangerous biological weapon ever released on the public is SARS-CoV-2. Wow. Now, let me go to smoking gun, which number 5? Smoking gun 5? Smoking gun number 5. Fauci's emails. Wow. Fauci's emails say this. Well, first of all, we must note Fauci's vacillations. When he was called, recently he was called to testify to the U.S. Congressional Committee, and this is all on video record. He said, no, U.S. taxpayers did not fund the Wuhan laboratory. Then he said, yes, U.S. taxpayers did fund the laboratory, but it was a sub-grant. Then he said, we funded it, but there was no gain of function research. Then he said, we did fund it, There was gain-of-function research, but it was a sound scientific decision. Then he said it would have been negligent not to fund it. So he kept vacillating back and forth, and I said, this is all on record. Let me go further to another one of Fauci's emails. This is from Dr. Christian Anderson, and I'm proving to you that SARS-CoV-2 is a genetic bioweapon. Dr. Christian Anderson, in an email, a secret email that was unearthed when Fauci's emails were exposed, he said the unusual features of the virus make up a really small part of the genome. So one has to look really closely at the sequence to see that some features look engineered.
Wow.
Smoking gun number 6. Further, this is the same guy, Dr. Christian Anderson, writing in an email to Dr. Fauci. Further, I should mention that after discussions with Eddie, Bob, and Mike, his fellow virologists, we all find the genome inconsistent with expectations from evolutionary theory. This is what Dr. Gill was referring to. The way a virus normally evolves, this virus didn't look like that. So they found, number one, the virus looked engineered, and it looked inconsistent with evolutionary theory. Wow. So, beloved, this states very clearly that SARS-CoV-2 is a genetic bioweapon. And let's go to Charles Leiber. Charles Leiber is the head of biology and the head of chemical biology at Harvard University. He was. And this guy used to train graduate Chinese students. Yes, that's him up on the screen. Used to train graduate Chinese students in his lab. He's a microbiologist of the highest note, and he has received over $15 million in defense contracts from the United States government to do just that, create genetic bioweapons. Two of his students were arrested at the Logan International Airport in December 2019, smuggling 21 vials of sensitive biological material to Wuhan, China. Amen. Charles Leiber himself was arrested by the FBI on January 28, 2020 for espionage with China. He was their consultant and actually was a hands-on builder of the BSL-4 lab in Wuhan, China. China has one deluxe BSL-4 lab. It was built by Charles Leiber through his consultancy. He was paid $150 million to do that and had a salary of $50,000 US a month from China. Amen. And so it has become very clear that SARS-CoV-2 is not a Kung Fu virus or a Chinese virus, as some people say. It was due to Anglo-Asian complicity. Amen. Leiber's involvement actually goes much deeper than I mentioned in, but that's not for this paper. I will skip at this point. I have talked extensively about vaccine types and mass vaccination at the time of a pandemic. But I'm just going to hit on one thing quickly before I go to my final point. Yes, I want you to understand that the vaccine types that are on the market, Pfizer, Moderna, AstraZeneca, and Johnson & Johnson, are all genetic vaccines. They are mRNA and viral vector vaccines that will affect your human genome. They are not normal.